It's a pleasure to be here as well. Thank you so much for the invitation. It's such an honor, a pleasure to meet the family finally, to meet you, Dr. Rosenkranz, in person finally. It's really a great pleasure. So my name is Andres Moreno. I am the winner of the award in the second year, back in 2012. I am from Mexico. I'm born and raised and trained. I went to medical school in Guadalajara in Mexico. Then I moved to do grad studies. I went to Spain, Barcelona, to do a PhD in population genetics and evolutionary biology. Then I moved to the States, so I did a postdoc both at Cornell and then here at Stanford with Dr. Carlos Bustamante, who's also very well known in the field of genetics. So, and then I was appointed a research associate, so I was here at Stanford for, at the end I think it was like for 5-6 years. And recently I moved back to Mexico to set up a whole new lab. And I will tell you about the adventure that has been this whole journey. So let me start by saying that you may have heard this story about this guy who's a Latino student of Mexican descent that just got accepted to go to college here at Stanford. Turns out that he went to the dentist for a regular check, and he shared the great news with the doctor that he got accepted into Stanford, who questioned his ability to qualify for Stanford, assuming that his acceptance was mostly due to his economical disadvantage and his minority status, right? So the story went viral, and I found it quite interesting because actually all of my research is focused on minorities, right? Particularly from Latin America. Myself, I was born in a rural area in southern Oaxaca, within a Mixteco community. And from earliest stages in my life, I appreciated and realized the richness of diversity that surrounded ourselves in Mexico and Latin America in general, as you can see here. So we clearly can detect that people is of mixed origins. We have all sorts of backgrounds, different stories. And such richness is actually more complex in Latin America because of the mixture of stories that get together in a single territory. And this is mostly due to recent population history intercontinental exchanges from the past 500 years after the European contact, and soon after that, the importation of African populations into the continent. So genetically, we can now use tools to study this diversity, and I think it's, again, a very rich source of opportunities to understand where we come from, what makes us different populations in each region of the world. Despite this richness, the problem for medical genomics is that up to 96% of the participants of the current studies in genetic association studies are from European descent. That percentage is changing, hopefully. I mean, that's the good news. But still, we are quite low in the percentage of participants that come from diverse populations from the rest of the world. So that was the main motivation for our group to devote our research into underserved and understudied populations throughout Latin America. But why is that important for medicine, right? So let's imagine this example of a patient goes to the doctor and has a disease that it has some genetic component, and the doctor says, well, it's all in your genome. Yes, I mean, it might well be true, right? There's some genetic factors there that are affecting the disease. But we're looking at the surface, right? And there are genetic variants that have different origins, right? That have different backgrounds. And the reason why evolution is important in medicine is that because if we do not understand the origin of those variants, right, we will not be able to deploy different measures and public health measures in different regions of the world. So that's what has motivated basically our efforts from the past 10 years. And that coincides with the time I was actually supported by the Rosenkranz Award. I was working in different projects that have regional approaches throughout Latin America. So I would like to mention 3 of them. So the main focus has been, like I said, region by region. We have a study focused on Mexican diversity that was published in 2014. Then we focused on Caribbean populations, and another one on South America. So for example, in the Mexican diversity project, we studied up to 20 different ethnic groups from throughout the territory. We generated sequencing and genetic data from these individuals. And we created a network of genetic connections between different groups. This is indigenous groups only within Mexico. So we can see here that most of the indigenous groups are actually quite isolated. Most of the connections happen within populations and very little connections between different populations that are far away. So that told us about the great degree of differentiation within a single country. So it's not the same being originated from the northern part of Mexico than the Yucatan Peninsula, for example. But not only that, we also found that there are differences in physiological measures that have clinical clinical relevance between people, for example, in the south, in Yucatan Peninsula, from the north. And this was just, to give an example, found by measures of lung capacity, for example. There's different lung function measurements that were different from people in the southern part of Mexico, only due to the origin of their indigenous component in their genome. It was not due to geographic differences or sex or age. It was due to the type of indigenous ancestry they have. So if you have Mayan genes, you have a different lung function than if you have, for example, Tarahumara genes or Seri genes from the Sonora Desert. So that was important to know that even the geographic distance is short, the genetic differentiation may be high, and that has biomedical implications. So that study got published in Science in 2014, 2 years after being supported by the Rosenkranz Prize. And this was a collaboration with the major genomic medicine institute in Mexico, INMEGEN, and jointly with Stanford University. But we wanted to deploy and apply similar strategies in other parts of Latin America. So for example, in this study in South America, we studied, again, some reference from Mexico, but also we included different countries from South America. The first thing you can see here, and this is just the principal component analysis of the genetic signature you find from each country, you can see how there's no overlap between the genetic signature from any country in South America and Mesoamerican Mexicans, right? Meaning that this is important for genetic variants that may not be shared between the two regions. And if we keep considering this Latino population as a single homogeneous group, we may be wrong. Even within South America, you can see, even though there's some overlap between countries, see, for example, Peru and Colombia. Despite being neighboring countries geographically, they do not overlap in the genetic signature, with Peruvians being more similar to Andean, Quechua, and Aymara, and for example, Colombians being closely related to the other Amazonian groups from eastern South America. So we wanted to go even at a finer scale and try to approach in a similar way we did in Mexico in a national diversity study, for example, within Chile. So we started a collaboration with Chilean researchers in this Chile Economical Project to try to understand what are the differences of the Chilean population in this, I wouldn't say 3D axis because Chile is just like one strip, so maybe just two-dimensional geographic space, but to try to understand what is the difference in the population. So that project led to another extension, which I found very interesting. I would like to talk about very briefly. This is a project that also is in a Chilean territory, but geographically and historically is very different, and this is Easter Island. So we had the opportunity to actually start a whole independent project in Easter Island, completely funded by the Rosenkranz Award. And I found it interesting because Easter Island is actually the most remote inhabited place on Earth. So it's, you have 6,000 kilometers from the South American coast or 6,000 kilometers to the closest airport to the Eastern Polynesia. So here there's admixture as well, like in Latin America, but here the origins are different. We have Polynesian ancestors on one side, and recently we have European admixture, and because it's part of Chile, we also have genes from Latin American origin. So we have, again, a scenario in which we could apply our tools, genetic and bioinformatic tools, to understand what the diversity in the island. So we went there, we approached the community, we gave talks, we invited participants, we took samples, measurements, and this is the sampling team back in 2013, which included so the anthropologist of the team, who is my wife, and also our daughter here, who was not even 1 year old back then. She was also taking measurements and samples. We collected samples, we went back to the lab, and what we do bioinformatically now is that we take the genetic data, and we are able to estimate at each single part of the genome, and this is the 22 chromosomes of the human genome represented by this line, where that site in the genome comes from. It could be either of Polynesian ancestry in green, or it could be of European ancestry in red, or Native American in blue. So most of the participants look like that, like a mixture of different ancestries in a single genome. And we wanted also to give back these results to participants, not as a percentage of how much you are from Native American or Polynesia, but just like a picture of yourself in terms of diversity. So that's what we did. We gave them just this picture without any figure of exactly how much Polynesian you are, and they were actually very surprised to either confirm what they know from their own family history, or to say, well, you know, I knew it, we are mostly Polynesians, so we should go to the Chilean government and say that we are not part of Chile, right? We don't know what happened after we left the island, so maybe now they are claiming independence or something. So more scientifically, what we want to answer is we want to deploy measurements of how different are the islands across the Pacific, not only Easter Island, but where Easter Island is located in the genetic space in the rest of the Pacific. So we're trying to get data from this gap that nobody has studied before at the genomic level, from Easter Island to the rest of the island, from remote Oceania. So that's what we're doing now with collaboration with the University of Oxford, for example. We are, I think, the only laboratory in the world that is sequencing genomes from Easter Island and Polynesia. We are already having preliminary data that shows that Easter Islanders have a genetic component that is not shared across any other island into the Pacific. And also we are trying to understand where they came from, because it's another thing that is an open question. Nobody knows where the initial settlers from Easter Island came from. Even though we know it's from Polynesia, we don't know exactly from which island. So let me finish by saying that most of this work was actually initiated here at Stanford, at the Center for Computational Evolution and Human Genomics, founded by these two brilliant professors, Carlos Bustamante and Mark Feldman. Also, thanks to their support, all these projects were possible. But also, with the Rosenkranz support, I was able to generate new data to start independent projects in my current position, which is now in this new laboratory in the state of Guanajuato, located right there in the center of Mexico, where I am the director of the genomics core facility. Basically, I am running the largest sequencing facility in Mexico and possibly across Latin America, one of the biggest ones. We have all different platforms for sequencing, genotyping, and basically helping to foster genomics in the region. But I also established the first research lab in Mexico that is focused on human evolutionary genetics. And I think that's quite novel because it's a new discipline. And a reflection of that is also the funding landscape of our lab. And if it wasn't for private initiatives like the Rosenkranz Prize, for example, I wouldn't have any startup. I wouldn't have any way to start a lab. So thanks to the Rosenkranz support, I have several projects. I have talked about the Latin American genomics one. I've talked about the Pacific project. But as you can see, the other agencies are either, for example, from the UK. We recently got awarded one of the biggest grants ever in population genetics in Mexico from the Medical Research Council from the UK to do the Mexican Biobank. Or, for example, we have another project funded by this International Center for Biotechnology in Trieste, Italy. But none of them is solely funded by the Mexican government agency. So we need a hard work to do to convince authorities in Mexico that population genetics is important for medicine. Which is why I think that the Rosenkranz Prize, for example, is a really clear, important step forward to demonstrate that we can do cutting-edge science in developing countries that is of international relevance. With that, I will really thank you so much for the opportunity to work in this field.